Hello, everyone.
Hello.
Welcome to class. See if I can get a little more light on the situation here. I hope everybody's doing well today. We are waiting on a few people, so
I have a quick question.
yes.
So for our first lecture video, was the introduction to microbes the only one that we had to watch, and for the lecture quiz?
That's right.
Okay.
This week there's only one lecture topic, and that's just, it's the first week of class. So
Right.
typically we have two lecture topics a week. So you'll have two lecture quizzes a week. But this week, just because it's the first week of class, we took things a little more slowly.
Gotcha. I only asked because the video description underneath it says number 1 of 4. So I just wanted to make sure there wasn't like 3 other parts.
So there are, I split all of the lecture videos into short, like 20-minute, 25-minute videos. So the introduction to the microbes lecture is made out of 4 videos.
Okay.
So one will be called 1 of 4, one will be 2 of 4, 3 of 4,
Okay.
4 of 4. Yeah. That's all I meant by that in the description.
Thanks for asking that, Brooke, because I was wondering.
Yes. I used to post the lectures as, in their entirety. So they were typically 60 to 90 minutes, depending on the lecture. And I had some trouble with YouTube in terms of getting those videos processed. So now I just divide the lectures up into shorter ones. So good question. Hold on just one sec. I'm going to adjust something here. See if we can get this a little clearer. Like this. See if that makes a difference. Not quite. Okay, that's a little better. It's a little gloomy out today, and as you can see, I'm filming in my home, so the lighting can be a little challenging for me. All right. We're still waiting on a few people, but I'm going to go ahead and get us started. Why don't you go ahead, if you would, and just type your first name for me in the chat. So I have a record that you came to class today. Thank you. That's great. Yeah, we'll do that at the start of each one of our meetings. And we will spend the first few minutes of each meeting just checking in with everybody and seeing if anybody has any questions like you've been asking, which is great. This is a good time if you have any questions about technical things or scheduling things, feel free to ask. And as always, you can type questions into chat, or if you'd rather, you can unmute yourself and ask a question out loud. That's fine too. What we will do, unless you need to speak to us, is I'll have you go ahead and mute yourself and also turn off your video feed. That will help us to stream more smoothly. We're a big group in here, so it just makes things go more smoothly. We have fewer times where the feed will freeze up if we go off of audio, off of video. And then again, when you want to speak, or if I call on you and ask you to speak, you can turn your video back on, you can turn your audio on. It's not that difficult. And that way, hopefully we won't have any streaming freezes and drops and things like that. I will say Zoom is getting better and better with time. Back in the spring, it was a little more unpredictable. but it seems to be improving with time. So that's good, because I know everyone is depending on it right now. Everyone is using it right now. So, all right, good. Yeah, so if you're just coming in, go ahead and type your name into the chat for me. That'll be my record of attendance that you've been here. Very good. So the last time we were together, we talked about how the course is organized and how we're going to run things this semester. I did have a couple of questions over email. So I just want to reiterate to make sure everybody's clear that we are doing something very unusual right now with scheduling. When you signed up for the course, you signed up for a lecture period and you signed up for a lab period. Everybody takes the same lecture time, and that's the 11:20 to 12:30 time slot, Monday, Wednesday, Friday. But everybody signs up for different lab times. So in order to get everybody together in one group for these Zoom meetings, we decided to use the lecture time for lab, for Zoom lab. And of course, your lecture, you're going to be able to do whenever it fits your schedule. You're going to be able to watch the narrated lecture videos whenever it fits your schedule. So each week we're going to meet Monday, Wednesday, Friday. Occasionally there'll be a day we don't meet. You can find those days on the schedule, and I'll try to remind you of those days. But for the most part, plan on meeting Monday, Wednesday, Friday on Zoom from 11:20 to 12:30. We won't be meeting at any other time. So if you signed up, let's say for the lab period that's meeting this afternoon, I think it's like 12:40 to 2:30 or something. We won't be using that time slot to meet on Zoom. We won't be using any other time slot to meet on Zoom. Just Monday, Wednesday, Friday, 11:20 to 12:30. Okay. And we're going to use this time to do our lab exercises because we want to be together and doing the labs as a group. I'll be able to do some demonstrations for you as we move through the semester, and we'll have the opportunity to do lab as a group. Okay. When you go on to Blackboard and you go into the, you click on the link in the menu for Zoom lab meetings, you should be able to see our scheduled meetings. And there'll be a button there for you to join the meeting. That's the best way to come into a meeting. It is possible to come into a meeting just using the Zoom app. You just have to have your meeting ID number and our meeting password. And those two things, our meeting ID and our password, stay the same the whole semester. And you can find those in our welcome announcement message when you come on to Blackboard. But like I said, the easiest way to get into these Zoom meetings is to get on Blackboard, click where it says Zoom lab meetings in the menu, and come in that way. I do typically, at least on Monday and Wednesday, I do typically use the whole lab period. So on Mondays and Wednesdays, plan for us to go all the way to 12:30. There will be some times when we don't. We may not go that long today. I don't really know. But many weeks we will. The lab period that I don't tend to use fully is the Friday one. So we may end up on Fridays finishing up a little early. Just so you know, looking ahead. And, I recognize that things happen, and I know some of you have children at home, and sometimes children are home unexpectedly, and sometimes you have to you're watching a child, or you have a sick child, or you have a sick family member or partner or roommate, and you're kind of frazzled and trying to do multiple things at once. I absolutely understand that. I just ask that you let me know if you have to miss one of our lab meetings, one of our Zoom meetings. Just send me a message through Blackboard. I post, well, first of all, Zoom records all of our meetings and they send them to me. And then I post them on our YouTube playlist. So if you have to miss a lab, it's not the end of the world. You can watch our meeting in a video when it becomes convenient for you. You can also go to those videos just if you want to listen to something again. I know I'm going through the lab, and hopefully you're taking in a good set of notes for yourself. But sometimes you miss things, so you can always go back and pull up our lab meetings on our playlist, and you'll be able to review something maybe you didn't hear the first time. Okay. So does anybody have any questions about how things are scheduled or when you're expected to come onto Zoom or anything like that? Anybody have any questions about that? And just so you know, if you ever accidentally click on the button that puts you into a Zoom meeting, if you ever enter a Zoom meeting accidentally, like you don't really want to, but you did, you have to exit back out. You have to leave the meeting or whatever, or Zoom will have you in the meeting, quote unquote, and Zoom will keep buzzing me, telling me, oh, so-and-so is in the meeting waiting for you. So just so you know, if you ever accidentally enter a meeting that you don't mean to enter, be sure that you then exit that meeting, leave the meeting, or else Zoom will like watch you and worry about you for forever, probably. I don't really know. Okay. So, okay, so I'm getting a message. You have to forgive me. First of all, I don't have my reading glasses on and my eyesight is terrible, but I'm seeing a message. I'm confused. Oh no, I don't want you to be confused. And the question says, so we have class at 12:42? And the answer is no. We meet on Zoom only 3 times a week. Monday, Wednesday, Friday, from 11:20 a.m. to 12:30 p.m. That's it. So even though you may have on your course schedule, you signed up for a lab period, Monday afternoon or Tuesday afternoon or Wednesday afternoon, we're not going to use those time periods. Okay. Now, the reason it still shows up on your schedule is because we want to give you credit. So you have to have on your transcript, you have to have that you attended both a lecture and a lab, which is why you're still signed up for both things at separate times, because we want to give you credit for attending both things. It's just that we're using our lecture period for laboratory this semester, and we're doing the lecture asynchronously, not on Zoom. We're doing the lecture outside of a scheduled class time. Okay, I know it's confusing. This is one of the difficulties of remote learning is all this scheduling stuff. But in the system, you have to be registered for both a lecture and a lab so that you get credit for both things. Oh, I have a good question from Amelia about our lecture this week, Intro to the Microbes. And Amelia's asking, what is the biggest defining factor between bacteria and archaea? I'm not sure exactly what you mean by defining, Amelia, but bacteria and archaea are 2 separate domains, remember. Every living thing on this planet can be organized into one of 3 groups. It's the most overarching classification is called the domain. So there's bacteria, They evolved first. They were the first living cellular life form on the planet. So they're a domain. Then come the Archaea. They evolved later. They are their own group. And then lastly came the Eukarya. And you and I belong to the domain Eukarya. The thing that unites bacteria and Archaea is that they are both considered prokaryotes. When you hear that word prokaryote, what you should think of is single-celled life form, no organelles in the cell. Both bacteria and Archaea are prokaryotes. They are single-celled life forms, and they have no organelles inside of them. So they're very primitive cells, structurally speaking. Now, the Eukarya domain contains eukaryotes. That's easy. Those two go together pretty easily. And what makes a eukaryote is that they can be single-celled or multi-celled. Both things exist in eukarya. You can be a single-celled eukaryote, you can be a multiple-celled eukaryote, but all eukaryotic cells have organelles inside of them. Nucleus, endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, membrane-bound compartments inside of their cells that do certain jobs for the cell. That's what makes a eukaryote different from a prokaryote. And both bacteria and Archaea are prokaryotes. Good question. Sometimes that language is very confusing, and it's easy for students to confuse domain with the classification prokaryote, eukaryote. All right, very good. Other questions? Other questions about that first lecture? Remember, while you're watching that lecture, you're going to want to take a nice set of notes for yourself, because you're going to be able to have those notes by your side when you take the lecture quiz and when you take the lecture exam. So make them the way you want them, okay? As complete as you need them to be. I do recognize that for some of you, some of this introductory material, you already know. It's already well known to you. But to others of you, this may be something you haven't seen in a long time, or it may be something you've never learned. So be sure that you have a nice set of notes taken for yourself so that you have all the information you need.
You made a note on your PowerPoint slide saying with the asterisk, bacteria and viruses, and then you said that they are acellular and not alive. Yeah, if you want to just explain just a little bit on that for me, please.
So bacteria are living organisms. They are cellular. They are single-celled organisms, but they're cellular. And that means they have a certain set of characteristics. We talk about that in a lecture called The Cell, which you'll be watching next week. All cells, for example, have a membrane around them, and all cells can reproduce themselves, and all cells metabolize. So there's a certain list of things that all cellular life has in common. Here's the thing about viruses. Viruses are acellular. They are not cellular. And by definition, that means they're not alive. Now that really freaks people out when you tell them that. Viruses do not meet our definition for living things. They are lacking in some of those key features that living things have. probably the most important thing is that viruses cannot replicate themselves. That's a feature that we have said all living things can do. It doesn't matter if you're part of the bacteria domain, or you're part of the Archaea domain, or you're part of the eukarya domain. If you're a living cellular organism, you have the ability to propagate yourself, to copy yourself in some way or another. So what makes viruses unique is that they can't do that. We call viruses particles because they are not cells. They don't have the things that cells have. Viruses are basically inert until they get into a living cell. So let's think about this SARS-CoV-2 virus, the one that's plaguing the world right now. This is a type of virus called a coronavirus. And you've all seen the picture. I know you have, because like every news outlet uses the same picture of SARS. It's a ball, right? And they always draw these red spikes coming out of it. Well, first of all, they're not really red. Someone has colorized that image. But let me tell you what that ball is. It's a piece of genetic material, and it happens to not be DNA. It happens to be RNA. And around that genetic material, there's a coating of protein. And sticking out of that coating, there are spikes made out of protein. It's the same protein that you have in your body and I have in mine. It's just a much smaller piece of protein. That protein spike matches a receptor, a protein receptor on your cells, some of your cells. And it fits like a key in a lock. That's the analogy people always use. The spike protein fits into the lock that's on some of your cells. When the spike goes into the receptor, the viral particle ejects its genetic material into the cell. Now, the cell doesn't know that that's viral RNA. It just looks at it and thinks it's RNA. And it does what it always does with RNA, which is it uses it. It uses it to make proteins, and it makes copies of it. And what ends up happening is new viral particles are built inside of that host cell. And eventually they will exit that host cell and they'll go on to enter other cells. So the best way to think of a viral particle is it's a machine. It's not alive, it's a machine. It's a protein machine. And it has instructions inside of it that living cells can use to make more viral particles. And that's what happens. Your cells get hijacked by viruses. But there's no, you'll hear me say, especially when we talk about viruses in a later lecture, you'll hear me say, there's no purposeful interaction between a virus and a cell. The virus isn't thinking about surviving. The virus isn't trying to keep itself alive. It's just a piece of protein that fits into a receptor on your cells, and your cells just make copies of it because that's all they know to do. So viruses are very unique. We still consider them microbes, but they are not living organisms because they are not cells. It always freaks people out when you tell them that. All right. Very good. Okay, any other questions? Okay, so what we're going to be talking about today is biosafety. which is just a very general topic, laboratory safety, basically. But we're specifically going to talk about biosafety because in a course like this, we're talking about laboratories that work with biological materials, that work with microbes. So we're going to talk about the best practices that we use in microbiology laboratories. A lot of you have indicated that you're interested in going on in your professional life to work in the healthcare profession. Many of you are going to end up working in hospital spaces, and you will end up moving in and out of laboratory spaces as a result. Sometimes it might be a diagnostic lab or a pathology lab, or you may end up going in and out of a research lab in a hospital. And laboratories have certain rules and regulations applied to them in order to keep us all safe. So even though we won't be going into the lab space together in person this semester, we're going to learn general biosafety. And then the other thing we have scheduled for lab this week is care and use of the microscope. That will be what we'll talk about on Friday. Before I start into our lab topic for the day, though, I just want to pull up our YouTube playlist, just in case anybody's having any difficulty there, finding it or getting on to where they need to go. So bear with me just a minute. I'm going to pull up first of all, our YouTube. Let's see if I can find it here. Okay, there we go. And now I'm going to share my screen with you if all goes well. You never know. There. All right, so hopefully what you're seeing right now on the screen is our first lecture topic, which says Introduction to the Microbes. But if you look up into the corner of the screen, you should see that we're on YouTube. See where it says YouTube Premium? So what you're looking at on this page is our playlist. We have a playlist set up on the YouTube channel called Science Science. And this playlist has videos just for us in it. Right up here, you can see the name of the playlist, NEC by 3210, I can't speak today, microbiology. That's our playlist. So when I gave you two web addresses, I gave you this, let me pull this out. I gave you this web address. This is the address just to the channel. All right, so the channel's called Science Science. And as you can see, if I click on videos, this channel is loaded with video material because I teach so many other courses. All right, so rather than have you search through all of those videos all the time, if you click where it says playlists, you can find our playlist, which is the NEC Bio 3210 microbiology list. And here's where you're going to find all of our videos, all the videos that I assign you to watch. So you can see here's one that says Intro to the Microbes, number 1 of 4. That tells you that this lecture has been split into 4 shorter videos. Here's the first one, here's the second one, here's the third one, here's the fourth one. Then underneath that, you can see a recording of our first lab meeting, which was Monday. So you can see that this one says lab course intro, February 1st, 2021. So this is a recording of Monday's lab. And then I've also posted next week's videos for you because remember, I try to post videos about a week early. You don't have to watch them right now. If you go to the schedule in the syllabus, you'll see that you don't have to watch History of Microbiology till next week. But it's up here for you just in case some students want to watch it early. You can see this is also broken into 4. So there's 1, 2, 3, 4, History of Microbiology. Then here's that lecture called The Cell, which is also for next week. There's 5 videos for that one. Okay. So I will post for you on YouTube on our channel about a week in advance. I know some students like to click on notifications for that for YouTube. And so they'll see when I post videos. Just don't feel like you have to rush and watch when a video gets posted. Check the schedule and see when you're supposed to be watching it to take the quiz on time. And if you want to, you can watch it early. And if you don't want to, you don't have to. As long as you get the quiz into me on time for each lecture topic, you're fine. Okay. All right, very good. All right, I'm going to share the screen again with you, and we're going to move into, if I can manage this. Okay, hold on just a sec. Come on, Zoom, let me do. Hold on just a second again. I'm going through 1,000 clicks here. Share screen, here we go. All right, now what you should be seeing on your screen is a PowerPoint slide that says biosafety in the microbiology laboratory. If you don't see that, unmute yourself and give me a little shout. But that's what you should be seeing now on your screen. So most of what I'm talking about today, I'm talking about labs that handle microbes. But a lot of what I'm going to say, you can apply to any lab, including what's on this first slide. Anytime you walk around in let's say a college or a commercial business that does laboratory work, or a hospital, or any kind of a place that has working laboratories in it, one of the things you're going to notice is that there's a sign on the door to the laboratory. And these door signs are universal. You can see over here on the right side of your screen, this sort of diamond label, a diamond shaped label, and it has 4 colors in it. And I know it's hard to see from where you're sitting, but the red box is there so that we can communicate any fire hazards that are in the laboratory. The yellow box is there so we can communicate about any chemicals that are very unstable in the laboratory and might cause problems because of that instability. The white box is there for very specific, very unusual hazards in the lab. And the blue box is there to communicate any health hazards in the lab. So all laboratories are required to post this on the doors. And it's there so that you can know whenever you walk into a lab, what hazards you are walking into, essentially. Because all labs are not equally hazardous to you. Some labs have a very low hazard, and some labs have a very high hazard to them. In fact, there are many lab spaces that you would not be able to just walk into without the right credentials. So the way that this diamond appears on the door is there will be numbers in each one of these boxes. And those numbers range from 0 to 4, with 0 being the lowest level of hazard and 4 being the highest level of hazard. So if you walked up to a laboratory and there was a 4 in each one of these colored boxes, you would know that that lab had a lot of hazardous things in it. either there are hazardous materials in it or they are doing work that is quite hazardous, and you'd have to take appropriate precautions. Now, outside of these door signs and this labeling, there are also some things that may or may not be posted on the door, depending on where the laboratory is. But these are also considered universal for all laboratory spaces. And I have them listed here on the left-hand side. No food or drink in laboratory spaces. All right, this is universal. You are not to bring food or drink into laboratory spaces. You know, one of the things that I've noticed across time when I watch television shows and movies and things is that they always like to show lab people eating in their labs. And sometimes they try to do it in the most disgusting way. Like they'll show a coroner or a medical examiner standing over a dead body and doing an autopsy while they have like a ham and cheese sandwich in one hand. Okay, that never happens. Okay, that's just not allowed. It's not considered good laboratory hygiene or good practice in a laboratory to eat or drink. And that's because there are chemicals and agents and cells inside of labs that can harm us. So it would be a terrible idea to consume something that might be contaminated. So we just don't do it. We set aside other spaces at work where we can eat and drink. Now, it also means that you don't want to apply things like makeup or lipstick or lip balm when you're in a laboratory, because as you know, sometimes those things get into your eyes or they get into your mouth, and you could, again, be exposed to things that are hazardous to you. So, good practice is if you need to adjust your makeup or put anything on your lips, that you just step out of the lab to do that. Maybe go down to the restroom to do that. Generally speaking, it's just a bad idea to handle your face while you're in a laboratory because your face has places on it that enter your body. So we try not to touch our eyes, we try not to touch our nose or our mouth while we're in a laboratory. It's just not good habit because you could be, again, contaminating yourself with something. Next on this sort of universal list is to use gloves correctly. I'll speak more about that in just a minute. And next on the list says manage your waste materials correctly. You know, as people who are in a laboratory space professionally, we are responsible for handling materials safely, not only to protect ourselves and other people, but to protect our environment. So it's really important that we always know, that we're always aware of what we're handling and how we need to dispose of it. We don't want to throw things into the ordinary trash that could be dangerous to people or animals or the environment. So we have to be very careful about putting things down drains. We have to be careful about putting things in trash cans. We have to know where waste bottles are, hazardous waste bottles, and we need to know how to put things into them and what goes into them. We need to know what needs to be autoclaved. And if you're not familiar with that term, we're going to talk about that in this course. Autoclaving is a procedure that destroys hazardous microbes by exposing them to high heat and pressure. So when we have materials in a laboratory that are contaminated with living microbes, we put them into a special autoclave bag and we put them into the autoclave and we sterilize them. We kill off any of the living materials. And then that material can go in the trash. One more universal laboratory standard is down here at the bottom. Always, always, always wash your hands when you come into a laboratory to do some work, and wash your hands before you leave that laboratory. after you've done your work. Now, you as a professional, you may be going in and out of laboratory spaces, but not working in them. For example, it might be part of your job to drop off a patient's sample at a laboratory, but you may not actually do the analysis of that sample. So that may not apply to you. But any time that you are working in a laboratory, even if you're only going to be in there for 20 minutes running a single test, you wash your hands when you come into the laboratory, and you wash your hands when you're finished with your work before you leave. Now, the other thing to remember about when working in a laboratory or even going into a laboratory is that nowadays we often carry personal items with us all day long. I'm thinking primarily about cell phones, but also about things like backpacks and handbags and other materials, lunches, laptops, iPads, or other tablet devices. We carry stuff with us through the day now, and it's pretty normal for most people to do that. Those are your personal items. And in a laboratory setting, you have to treat those in a very particular way. When you arrive to work in a microbiology laboratory, you have to put your personal items away from your workspace. Now, in most laboratories, there is a place for this. There might be a little adjacent room to the laboratory. There might be a particular corner of the room, but there will be a place for you to sort of deposit your personal items. The reason you don't want your things in your workspace is because you don't want your personal things to get contaminated. You're not going to be washing your backpack when you leave that lab. You're not going to be washing your cell phone. You're not going to be disinfecting your purse. So you don't want it anywhere near your workspace. After you drop off your personal items and you're ready to get to work, really in any lab space, but especially in a microbiology lab, you have to clear and clean off your workspace. That might be just a little area, 2 feet by 2 feet on a bench somewhere, or you may have the entire bench to yourself. But the very first thing you need to do is clear it, number one, and clean it. And by clean it, I mean disinfect it. One of the things you'll hear me say again and again in here is that when you're working with microbes, you have to be careful not to contaminate your cultures of microbes. And you have to be careful not to contaminate yourself with your microbes. So you don't want to come in and start working on a bench somewhere that you haven't disinfected because it might be contaminated. So there are two disinfectants that you will encounter in most lab spaces that are used to clean work surfaces. The first one is alcohol, and the second one is bleach. Some lab spaces use only alcohol, some use only bleach, some use both. It depends. It depends on the lab and what their preference is. But I have some information here that you should know. You should know, for example, that a solution of alcohol that's diluted between 60 and 90% is a very good disinfectant for surfaces. It's interesting, one of the things we've learned in terms of the general public during this pandemic is a little bit about disinfectants, right? There was a time early on in the pandemic when people were very concerned about cleaning packages and cleaning their houses very frequently and disinfecting against the virus. And there were questions about whether or not they should be buying pure alcohol or whether they should be buying dilute alcohol. And the answer is you want a diluted alcohol. It works better than pure alcohol does against microbes. So anywhere between 60 and 90% will work. Most of the time, in my experience, we mix it up to about 70%. And that alcohol can be any of several kinds. Some laboratories use isopropanol, some of them use ethanol. It just depends on the laboratory. Alcohols are expensive, so some laboratories won't use them as disinfectants, but they're very good disinfectants. They work very quickly. The other very common one is bleach. And I'm talking about the same bleach that you might put into your laundry at home, the same chlorine bleach. But look, it's a very diluted solution. 10% bleach is a disinfectant and will kill the kind of microbes that we interact with in laboratory spaces. Either one of those is fine for cleaning your workspace at the beginning of the day and for cleaning up your workspace at the end of the day. Now, anytime we go to work in a laboratory, it's also really important that we gather all our supplies before we get started. I'll be talking about that this semester as we do laboratory exercises together over Zoom. The last thing you want to happen is for you to get started on an experiment and have your hands full and be doing some critical step in an experimental protocol, and you realize that you don't have something that you need. And it interrupts you. It may actually ruin the experiment. It means you're going to have to put things down and go get something, and it's just bad practice. So we always gather everything that we're going to need before we start working. Here's something else that's really important, and that's labeling. You'll hear me talk about this a lot too. In a microbiology lab, we purposefully grow microbes. We grow them in incubators, and we call those containers cultures. Sometimes we grow microbes on petri dishes, and sometimes we grow them in test tubes, but the idea is the same. We purposefully create a population of microbes in a container, and that's what we call a culture. Now, it's really important when we create these cultures that we label that container. And I've got the 4 things listed here that belong on any container in a microbiology lab. First of all, your name belongs on it somewhere if you made it. Either your name or your initials should be on it. The same is true if it's not a culture container, but let's say you're collecting a urine sample from a patient, or you're going to take blood from a patient, or some other diagnostic sample. Labeling that container is very important because that container is going to change hands. And it's important that everybody who touches that container knows exactly what's in it. Now, the nice thing in most hospital settings is that these labels are often generated for us by computers. But in a micro lab, we are making the container start to finish, so we have to label it ourselves. So your name or initials belong on it. The date that you made it belongs on it. The name of the organism that's in that container belongs on it. And what the material is in the container. So let's say you're going to make a culture on a petri dish. You're going to smear some bacterial cells on that dish, and you're going to put the dish in the incubator and let them grow. That dish has a semi-solid, almost like a jello material in it called agar. And agar will grow bacteria because it's got some nutrients in it that bacteria need to grow. But there are lots and lots of different kinds of agar. So it's going to be important when I make that culture that I tell what's in the agar. What kind of agar is it? Again, there's a chance that somebody else in that lab is going to encounter that dish, that petri dish. And they need to know exactly what it is when they pick it up, who made it, How old is it? What kind of organism is growing on it? And what kind of medium, what kind of agar is in it? So those are the four things that we always put on our containers in the laboratory. So there are lots of universal standards inside of laboratories, and those universal standards apply to all labs, including microbiology labs. There's also some general things that we follow that we refer to as lab hygiene. So again, this would apply to a microbiology lab or any other kind of lab. If you're working in the lab, you want to follow certain practices like like I said earlier, you want to keep your personal things out of your work area. You want to keep your work area clear and clean. You want to wear appropriate clothing when you're at work. Now, this is going to vary depending on what kind of lab you're working in. You can see in the picture over on the left, there's somebody working in a laboratory, and this person is wearing a lab coat. Lab coats are very common attire in laboratory spaces, but some labs don't have their employees wear lab coats. Some labs have them wear scrubs. In some laboratories, you will wear your street clothes. It depends on the laboratory that you're working in and what kind of work is being done. But you want to be sure that even if you're going to wear a lab coat, you want to be sure that the clothing that you're wearing from home is appropriate. In other words, you have to think about things like how flowy your clothing is. This primarily applies to women, but you want to avoid wearing shirts that are very long and flowy with long bell sleeves. Anything that might accidentally sweep by and touch your workspace, you want to avoid wearing. The same applies for jewelry. You don't want to wear necklaces to work in a lab space that are very long, that might accidentally drag across a desktop or accidentally touch a culture. You just have to think about those things and wear your appropriate clothing underneath whatever work clothing you're going to be wearing. You want to wear appropriate shoes. This is a really important one. Again, it's going to vary a little bit lab to lab, but the general rule is you don't wear anything that has open toes in a laboratory. No flip-flops, no sandals, no open-toed shoes in a laboratory. And that's because sometimes we drop things in the lab, and so we want our foot covered as much as possible. If you're like me and you have long hair, you have to manage your hair in a laboratory space, and that just means pulling it back and putting some kind of an elastic around it. And that's because just like clothing and jewelry, your hair can accidentally sweep down and touch your workspace. Your hair can contaminate your workspace, and your workspace can contaminate your hair. The other thing you have to think about with hair is fire. I can't tell you, I've heard at least half a dozen stories through the years of my time as a professional of people who set their hair on fire in laboratories. So it's a very serious issue. You've got to keep your hair back and restrained when you're working in a lab. The other thing, just in general, and I probably don't even have to mention this, is you always have to be very respectful of fire. We only set Bunsen burners and things on fire when we're using them, and then we shut them right off. You never leave a fire unattended in a laboratory, and you keep anything that's flammable away from that fire. Hopefully you all already know that. All right. I'm just going to stop here briefly and ask, does anybody have any questions about anything that I've said so far? You know, a lot of what applies in laboratory spaces applies just generally to people who work in healthcare. If you end up working in some role in the healthcare system and you're in a doctor's office setting or you're in a hospital setting, you'll be told what kind of clothing is appropriate. If they're going to give you clothing they want you to wear, you'll be told about your hair, what is okay and what's not okay, um, jewelry, things like that. And again, that's for your safety, and it's for the safety of the people you might be interacting with. You don't want what you're wearing and how you look at work to impact what you're doing on the job. All right, let's go back in here. I want to spend a couple of minutes and talk a little bit about PPE. That's another thing that's happened during this pandemic, is that the general public now knows what PPE is. PPE stands for personal protective equipment. And we use personal protective equipment in laboratory spaces, including microbiology labs. Now, there's all kinds of PPE. As I said, there's certain clothing that's considered PPE, lab coats and scrubs. There are safety glasses. There are more secure goggles that people wear over their eyes. There are respirators or face masks that people wear in laboratories. There are gloves. The kind of PPE that people wear depends upon the kind of laboratory they're working in. But, and here's a big but gloves are used in all laboratories. So put a little star next to that in your notes. It doesn't matter if you're working in a microbiology laboratory, or if you're working in a diagnostic laboratory, or if you're working in a pathology laboratory or a research laboratory. If you're handling chemicals or you're handling microbes, you're going to have gloves on. If you're handling patients, you're going to have gloves on. So gloves are the PPE that everybody deals with if they do any kind of laboratory work. And the thing about gloves is you have to put them on, you have to don them correctly, and you have to doff them correctly. You have to take them off correctly. And again, depending on the work you're doing and the things you're touching, that can be very important. You'll notice I have this sentence highlighted. When you're wearing your gloves, you don't want to touch anything while gloved that is not routinely disinfected. Let me say that again, because that's a really important point. When you're wearing gloves in a laboratory, and especially in a microbiology laboratory, you don't touch anything while you're gloved that cannot be routinely disinfected. So what kind of things are those? You don't want to touch your eyeglasses. You don't want to touch your hair. You don't want to touch your jewelry. You don't want to touch your laptop. You don't want to touch a microscope with your gloves on. Gloves, once they're on, should be thought of as being contaminated, even if they're not. Once you put your gloves on, you should treat them as though they have microbes on them. And don't touch anything that you cannot disinfect. Now, the other thing I've got listed here is don't reuse gloves. That's a terrible practice. If you need to take off a pair of gloves, because let's say you do need to touch something like your laptop, you don't ever put the same pair back on. You throw them out and you get a fresh pair. Listen, gloves can be expensive, especially if they're sterile gloves, and there are times when we have to wear sterile gloves. But we don't ever put a pair of gloves back on because, like I just said, we assume that the gloves are contaminated, and we don't want to contaminate ourselves by struggling with them to get them back on. You should also never leave the laboratory space with your gloves on. If you're working and you have gloves on and you need to go use the restroom, you take the gloves off. go to the restroom, come back, get a fresh pair of gloves, get back to work. If you have to step out into the hall to take a phone call, or you have to step over to send an email, step out of the laboratory, in other words, to send an email, you don't leave the laboratory with your gloves on. Now, what I want you to start to feel like, what I want you to think like, is that if I'm in a laboratory space and I'm working and I don't have gloves on, that's bizarre. That should just never happen. You get to the point with practice, and it does take practice, but you'll get to the point where if you ever did anything in a lab without gloves on, it would feel very strange to you. And that's good, because we need gloves to help keep us safe in the lab. One other thing I want to mention on this slide is laboratories are full of safety equipment now, which is a really good thing. But if you're working in a lab or you're taking a class in a lab, you always want to familiarize yourself with where that equipment is, so that if an emergency did arise, you would know where to get it. The last thing you want to happen if you're sitting in a lab is for an emergency to happen and you not know where the eye wash station is, or where the fire extinguisher is, or where the first aid kit is. So it's really good practice and a good habit anytime you work in a laboratory to know where all the safety equipment is. Many laboratories have eye wash stations now in case someone splashes a chemical into their eyes. Some laboratory spaces have emergency showers. If, for example, like I said a few minutes ago, somebody in the lab accidentally set themselves on fire, you can now push that person under an emergency shower and essentially save their life. There are first aid kits in all laboratory spaces. There are fire extinguishers in laboratory spaces. You don't want to wait for the emergency before you look around to know where the fire extinguisher is. So when we're in a laboratory, especially a microbiology laboratory, and we're getting to work, we want to have gloves on. Anytime that you are touching a culture, you must have your gloves on. Now, once you've handled a culture, even if you've handled it correctly, and you know that you didn't get any of that culture on your gloves, you still want to act as though your gloves are contaminated. So while you're wearing gloves, you don't touch your face, you don't touch your glasses, you don't touch your hair, microscopes, textbooks, cell phones, pencils and pens and computers. Door handles is another one. Don't touch things that are not routinely disinfected while you have gloves on. If you have to touch those things, you just take your gloves off, or even just take one glove off. It's a common practice in many different types of labs to do what's called clean hand, dirty hand. In other words, imagine if you're in a laboratory and you're doing an experiment or you're running diagnostic samples and you have to record the results while you're doing the work. It's very common for people to have a gloved hand that they can use to touch things, and an ungloved hand that they can use to record results. That's what we call clean hand, dirty hand. Another thing that you should know very generally about gloves, in a microbiology lab, gloves are considered regular waste. So even though, like I just said, we treat them as though they've been contaminated, we don't put gloves in the autoclave. We throw them away in the regular trash when we're done. Now that might seem strange to you because, we might have microbes on these gloves. The reason it's safe to put gloves into the regular trash is because microbes cannot survive on the surface of gloves for very long. So even if we did get some bacteria or even some viral particles onto our gloves while we were working, that particle or those cells will not persist on the gloves for very long. And so they're not considered hazardous and they get thrown out in the regular trash. One of the things we'll talk about in this course again and again is this. Microbes are all around us. The human body is covered in microbes, inside and outside. You are walking around right now as a healthy person, covered in microbes. Your skin is covered in microbes. Your mouth, your nose, your throat, your intestine, all of you, you are coated in a layer of microbes. And that's a good thing because the vast majority of microbes are actually helpful to you. They keep you healthy and they keep you safe. The other thing to remember is that the environment is covered in microbes. So even if you're a person that keeps a very clean house and you're constantly wiping down counters and things like that, your house is still full of microbes. And that's okay because most of the microbes that we encounter in this world are not going to harm us. We are constantly consuming microbes on our food, and that's okay because only a handful of them are dangerous to us. But you have to remember that in the microbiology laboratory, sometimes we're trying to grow pure laboratory cultures. We are purposefully trying to grow one particular microbe in a container. So we have to be very careful that we don't ruin that container by contaminating it with other microbes. Our body is covered in them, the environment is covered in them. So we have to keep our cultures safe so that they don't get contaminated with other microbes. You also have to remember in the healthcare profession that even if you've taken or a physician has collected diagnostic samples from a perfectly healthy patient, let's say a patient who's just come in for a physical exam, we have to always treat those tissue samples and fluid samples as though they contain microbes. Okay? And even though most microbes won't harm us, you never know when a pathogen will be present. So you have to handle patient samples as though they contain pathogens, and you have to be safe. So here's something I said earlier, I'll say it again. Perhaps the two most important things I'll say today that you have to keep in mind when you're working in a lab that handles microbes. Don't contaminate the culture with yourself, your body, and don't contaminate yourself with your culture. Don't contaminate the culture or the diagnostic sample with your own body microbes. And don't contaminate your own body with that culture or that diagnostic sample. Very important piece of information. All right, we're running out of time today, but I'm just going to continue on with a couple more slides and then we'll finish up on Friday when we get together. The last thing I'll talk about is this idea of biosafety level. Some of you may have heard this language before, and for some of you, it will be brand new. Whenever laboratories work with living organisms, so here I'm talking about microbial organisms, and I'm also talking about other kinds of organisms. So a pathology lab, for example, that deals with human tissue, or perhaps a diagnostic lab that works with human samples, or again, a microbiology lab that works with microbes. Anytime you're dealing with a laboratory that works with living organisms, living cells, it has to be assigned what's called a biosafety level. And we shorthand that as a BSL. These are standards that are applied to laboratories that are designed to keep the people working in the lab very safe, and also the general public. Now, I have a lot of information on this slide, but I want you to make a note for yourself that you do not need to know the list of standards for each biosafety level. I'm not going to test you on that. What I do want you to know is that as the biosafety level number goes up, that means that the laboratory is handling more and more and more dangerous living organisms. So for example, a BSL-1 lab is the lowest level of hazard. Teaching labs that are in colleges, for example, are BSL-1 labs. We handle microbes in our microbiology lab, but we don't handle any pathogens in there. That's a BSL-1. It has basic lab safety standards and basic PPE. If you go up to BSL-2, now you're talking about a laboratory that is dealing with certain pathogens, and particularly human pathogens. But these are pathogens that have very specific characteristics. They are non-respiratory, and if someone were to get infected with them, there are treatments available and or vaccines available. So a BSL-2 lab might handle, for example, an organism like E. coli, which sometimes causes a gastrointestinal disease, or an organism like Staph aureus, which can sometimes cause skin disease. Non-respiratory pathogens that have a treatment available just in case, or perhaps a vaccine available just in case. Now, what BSL-3 and BSL-4 are very similar. These are high hazard laboratories. There are lots of BSL-3 labs in our country at larger, usually state-run universities. There are very few BSL-4 labs in our country. You should know that there's a couple BSL-4 labs down in Massachusetts. The difference between these two labs, a BSL-3 lab can handle respiratory pathogens, but we have to have either a treatment or a vaccine available for that pathogen. So a BSL-3 lab could handle, for example, influenza, the influenza virus. Maybe they do experiments on the influenza virus. We don't have a specific treatment for influenza, but we do have a vaccine for it. BSL-4 labs are the highest hazard level. And as you can see, there's a long list of requirements for those labs, again, that you don't need to know. But you should know that BSL-4 labs handle respiratory pathogens that have no known treatment and no vaccine. So right now, any laboratory that is studying the pandemic SARS-CoV-2 virus, the virus that causes COVID-19, any laboratory that's handling that virus in our country or around the world is a BSL-4 laboratory. And they follow very strict protocols to protect the personnel in the lab, number one, and to protect the general public. All right, we're just about out of time. Does anybody have any questions so far? We'll talk a little bit more about these BSL levels when we get together on Friday. I want to show you some pictures just so you can see the kind of PPE that people have to wear when they work in a BSL-3 or a BSL-4. I've never been in a BSL-3 or 4 lab. I've worked in BSL-2 labs, and of course I've taught in BSL-1 labs. I've never been in a high hazard laboratory before, but I know people who work in them, and it's not easy. There's a lot of time spent every day just dealing with the PPE. And again, it's important because it helps keep people safe. Any other questions? All right. All right, if there aren't any questions, I'm going to let you go. Remember, you can find this video up on our playlist later on today. It does take a little while for Zoom to process it and send it to me, but it'll be on our playlist later today. Remember, you have that lecture to watch if you haven't already. You have a lecture quiz to take this week. You'll have laboratory homework this week, but you won't be able to do all of it yet. You won't be able to do all of it until we have lab on Friday. I will see you again on Friday at 11:20 here on Zoom. But if you have any issues before then, if you run into any problems or you have any questions, you can contact me over email, and we'll talk, okay? All right, everybody, I'm going to let you go. Have a good rest of the day.
Have a good
Bye.
day.
Thank you.
Thank
We'll see you
you.
next time.
Thank you.